So this work is mostly due to Divyanshi and Akshay in my lab and Begum in Esteban Mazzoni's lab down the road here in NYU. So my lab is interested in transcription factor binding specificity, and in particular how pre-existing chromatin can determine where transcription factors will bind. So let's imagine that you have a cell type where transcription factor is becoming active into that cell type for the first time. Of course, when the transcription factor is choosing its binding sites, it will be using the intrinsic sequence preference that it has. But in terms of which motif instances will become bound, pre-existing chromatin will have a role to play in determining that. So for example, a transcription factor might prefer to bind in sites that are already accessible in the pre-existing cell type that it became active in, or perhaps it forms some interactions with other regulators that are already bound in that cell type, or perhaps it does neither and it pioneers new sites. So my lab is interested in trying to pull apart these different effects, seeing how chromatin and sequence features together determine binding. And we're particularly interested in doing this in the context of systems like developmental systems and transdifferentiation systems, where we have factors becoming active from one time point to the next. So for example, we recently did a study where we're looking at two proneural transcription factors in mouse embryonic stem cells. So we're looking at ASCL1 and NeuroG2. Both of these are basic helix loop helix transcription factors. If you express one or the other in mouse ES cells, you get neurons out at the end. So we started this, these are quite related transcription factors. We started this study with just asking if they're functionally redundant. Are they having the same effect on the regulatory network in both cases? So it turns out when we look at the gene expression patterns that are downstream of both of these genes, there's a lot of similarities. There's a lot of genes that are similarly upregulated or downregulated by both of these transcription factors, but there are some factor-specific activities. And specifically, there are certain genes that are only upregulated by ASCL1, and these seem to be subtype-specific neuronal genes, like genes associated with cortical interneurons, serotonergic neurons, neurogenergic neurons, and downstream of NeuroG2, there are genes that are more associated with spinal motor neurons or sensory neurons. So we have these two similar transcription factors, and they're having different regulatory effects. So how do these two related basic helix factors have those different effects? So it turns out that they actually bind quite differently on the genome. Again, we've overexpressed these both in the same context, separately in the same mouse embryonic stem cells, but they seem to bind quite differently on the genome. There's only about 10% of the sites that one or the other binds are bound similarly by both of these transcription factors. So what's causing this differential binding specificity? Part of the answer is quite simple. They have slightly different E-box motifs that they like to bind. So there's a core difference in the central dinucleotides of these E-box motifs that ASCL1 has one preference, NeuroG2 has another preference. In fact, if we just look at these simple Kamer sequences, CAG-STG or CAK-ATG, you can see that their presence is quite discriminative of this differential binding, right? So this intrinsic difference in binding specificity explains some aspect of the differential binding, but it doesn't explain everything about differential specificity, because there's these Kamers are all over the genome, right? There's millions of copies of them on the genome. Only some of them are bound and many of them are not bound. So chromatin plays a role in specifying which instances of the motifs will be bound. For example, if we look back at what the accessibility landscape looks like in the 0-hour time point before these factors have become expressed, so these are pluripotent cells, roughly half of the binding sites for ASCL1 or NeuroG2 are already accessible in that pre-existing state. Roughly half are not accessible. So we're interested in trying to pull apart what features a factor is looking at a given site, or maybe overall as a property of the transcription factor when it becomes induced. How much information is coming from sequence? How much information is coming from the pre-existing chromatin? So we've developed this neural network architecture that's lets us do some aspect of this, try to jointly analyze sequence and chromatin features. So our architecture is similar to what was nicely introduced by Anshul already. It's a basic CNN RNN architecture, but we have two halves to this network. One half is just looking at sequence features, and one half is looking at chromatin features, prior chromatin features. So these are chromatin features like attack-seq or histone modifications or whatever you have from the cell type before you have activated the transcription factor. So for example, in our example here, we're training this network to predict the genome-wide binding of ASCL1 or NeuroG2 And we're looking at sequence on one half of the network, and we're looking at the mouse embryonic stem cell chromatin landscapes on the other half of the network. Now you'll see this architecture has a simple structure here. We're only combining these predictions from these two subnetworks at the very end. And that's quite deliberate. We're doing this for the purposes of interpretability. We'd like to be able to ask, for a given transcription factor binding site that's correctly predicted, how much information is coming from the sequence side and how much information is coming from the chromatin side. So let's we train this network on ASCL1, for example, and we can map out all of the sites on the genome that are bound or unbound by this transcription factor according to these two scores that come from these two halves of the network. So again, one axis comes from the sequence subnetwork, one axis comes from the prior chromatin side. So you can see that these sites, the bound sites, which are the orange ones here, spread all over this two-dimensional landscape. You can see a high density of sites over here on the high-scoring sequence side of things. And a lot of those turn out to be high-quality motif instances. There's a lot of information that we can pull out from the flanks of those motifs that are quite informative. Even just motif multiplicity plays a high role in driving a high score from the sequence side. Similarly, though, there's a lot of density up here on the high-scoring chromatin side of things, right? So these seem to be sites that are already displaying active histone modifications and accessibility in the pre-existing mouse embryonic stem cell state. You can see the feature of this landscape, though. We think that there's some degree of mutual compensatory effects going on here, that if a site has a high enough sequence score, it doesn't really matter what the prior chromatin score was, right? It's going to be bound regardless. If a site is in a highly accessible or a highly active region, you might get binding even if you don't have a very nice motif score, right? And this maybe is somewhat intuitive, the way we think about how binding works on a genome-wide basis. So everything I've talked about here for ASCL1 is also true for NeuroG2. Again, there's this sort of degree of compensatory features from sequence and chromatin. So just like we can break down individual sites into a chromatin score and a sequence score, we'd also like to be able to just measure transcription factors in general against one another. How much are they affected by prior chromatin as opposed to just their intrinsic sequence specificity? So to do that, we can do something quite simple. We can train one of our neural networks using just sequence information and see what the performance of the network is on held out regions of the genome. And then we can train the network with the full thing, with the sequence and the prior chromatin information, and see the gain in performance that we get when we give it, the network all of that information. So you can see for NeuroG2 and ASCL1, we get a substantial increase in performance, here just measured by recall at a fixed false positive rate. We get an increase in performance when we add chromatin into the model. And we get a similar gain in performance for both of these transcription factors, right? So they don't seem to be, I mean, the way we interpret this at least is that both of these factors are similarly influenced by the prior chromatin. We have as much increase in predictive capacity by introducing prior chromatin for both of these factors. Okay, so that's ASCL1 and NeuroG2. I want to bring you back here for a second just to continue the story here on a slide that I showed here where we have some sites that were inaccessible, that were not accessible, at least according to a TAC-seq in the ES cells. They become bound in a differential manner by ASCL1 or NeuroG2, right? So they were inaccessible. When you have these sites becoming bound, we should see them now becoming accessible in a factor-specific manner, right? And that's exactly what happens if we look at where ASCL1 or NeuroG2 was binding differentially. We see differential ATAC-seq signals coming up downstream of their binding. We also see differential signals of active histone modifications coming up over the same sites, again, after we see binding induced. So now let's imagine where we are. We have ASCL1 and NeuroG2 have bound differentially to the genome. They're establishing differential chromatin accessibility landscapes on the genome. So any factor that comes in after them is going to see a differential accessibility landscape depending on whether ASCL1 or NeuroG2 was active before them, right? And there's lots of factors like that because, remember again, we're getting neurons out either way here. So there's lots of neuronal regulator transcription factors that become active downstream of both ASCL1 and NeuroG2. But now, when they're active, they're coming into these chromatin landscapes that are different depending on whether NeuroG2 or ASCL1 were active beforehand. So we looked at 3 transcription factors here, BRAIN2, EBF2, 1CUT2. Again, they're downstream, they're activated by both ASC1 or NeuroG2. And what we want to know is how affected these different transcription factors are by the now established pre-existing chromatin landscapes that the proneural genes set up. So we can again train our neural networks to predict their binding across the genome. But now our prior chromatin landscape is the accessibility landscape that was established by one of the proneural genes, right? So we can train this with just 1/2 of our data. We look at the binding downstream of ASCL1 and the accessibility downstream of ASCL1. So when we do that and we ask our network how much information is gained for each of these transcription factors from the prior attack-seq, we can see here now that there's differences between how the different transcription factors look. BRAIN2 and EBF2 seem to gain quite a bit of information from prior accessibility landscapes. 1CUT2 barely gains any information at all. The way we interpret this is that BRAIN2 and EBF2 are more affected by the chromatin landscape, right? They're gaining more information from the chromatin landscape, whereas something like 1CUT2 may be more of a pioneer factor and is not that affected by where the accessibility landscape was when it became active. So remember, we can test this because we have binding data available for both for these factors downstream of both ASCL1 and NeuroG2. So our prediction here would be that these 2 transcription factors, BRAIN2 and EBF2, would be more differentially bound, dependent on the different accessibility landscapes that are established, whereas 1CUT2 will just do its own thing and bind where it wants to bind. And that's exactly what we see, or we see data consistent with that, I guess, at least. We see a lot more differential binding for BRAIN2 and EBF2 downstream of ASCL1 or NeuroG2 than we see for 1CUT2. So our model here is that we have these proneural genes that are similar in lots of ways, but they're establishing different binding patterns on the genome, and therefore they're establishing different chromatin accessibility landscapes or other chromatin landscapes. And then these downstream transcription factors might be influenced in their binding by this pre-existing landscapes that were established by these prior wave of regulation. So this is sort of a type of indirect cooperativity effect mediated by just accessibility, we believe. Okay, so this is described, the biology side of this was described in the paper earlier this year. Our neural network is still up on bioRxiv. And I want to thank everyone involved in the lab, especially Divyanshi and Akshay and Begum over in Esteban's lab. And we have jobs available. So thank you.